Hello and welcome back to Neurogeneious. This is Rhea, and today in our second episode, we'll be moving on to a more exciting topic that will properly set us up to discuss more advanced technologies later on. So, today we will be discussing neurogenetic diseases or disorders and how they function on a cellular and primarily molecular level. We will touch on a little bit of epigenetics and how that can progress and affect these diseases. And then lastly, we'll dive into the basics of gene therapy. So, before we go deeper into what exactly some examples of neurogenetic disorders are, let's just talk about some of the common themes we might see in neurogenetic disorders. So, speaking on these disorders, with great genetic factors, we can see that there are lots of repeat expansion mutations. So, what exactly are these? We know that our genome contains lots of genetic information in the form of genes, where it's consisting of numerous pairs of nucleotides. But in the case of repetition, we can see that there's a certain array or sequence of nucleotides that is repeated to the point a function might be impaired. And we can see this with Huntington's disease, where the HTT gene gets impaired after numerous CAG repeats or CAG repeats multiply. And then this results in the aggregation of the Huntington protein, which actually leads me to my next point. A lot of neurological disorders face issues with an aggregation or misfolding of some type of protein. So by impairing the protein structure, we directly affect their function and other nearby cellular functions. Another common impairment is something called oxidative stress, where the production of free radicals overcomes the antioxidants in your body that can counter them. And these extremely unstable free radicals are able to damage DNA, which is therefore known as oxidative stress. Another important thing to note is that epigenetic factors could also play a role in how diseases could progress or even stop in some cases. We know a lot in biology generally that structure often correlates to function. So therefore in epigenetics, when we are changing the structure of DNA molecules, we are impacting function because of some sort of environmental condition. We can see that these epigenetic changes occur in two major ways. One of the ways is the addition of methyl groups, or changing the tightness of DNA around histone proteins, which are essentially the proteins that allow DNA to condense. In general, epigenetics is an important factor because it demonstrates how environment can extremely influence disease. Now let's dive a little deeper into specific examples of neurogenetic disorders. First off, we have Friedreich's ataxia, which occurs due to a mutation in the FXN gene that results in muscle weakness, loss of control to movement, which is essentially the definition of ataxia, and then additionally some other various neurological symptoms. Moving on, I've touched on this one earlier, and it's also quite a famous one, but Huntington's disease is one of the bigger neurodegenerative diseases with a huge genetic factor. So we know that there are lots of symptoms associated with Huntington's. Those consist of movement issues, emotional turbulence, slurring of words, and then as the disease progresses and becomes more severe, we can also see lots of memory issues. Additionally, with that, we have Fragile X syndrome, which is definitely something more specific to our sex chromosomes, and these can definitely cause more cognitive and behavioral issues. Then lastly, there are also a variety of neuromuscular disorders that also have huge genetic factors, DMD and CMT as noted here for short. These diseases do have some significant differences, with CMT impacting the peripheral nervous system and DMD being more of a muscle degeneration issue. But yeah, those are essentially the major disorders and diseases that we can identify having a major genetic factor associated with them.
But you might be wondering why I didn't include other major diseases like Alzheimer's, Parkinson's, and ALS. And it's actually not because they don't have genetic factors at all. They do. But the extent to which the genetic factors have been researched and found to progress the disease is less than these other diseases I mentioned. ALS in particular actually only has 10% of their cases being derived through inheritance or genetically. The rest are, as we call, sporadic, which is 90%, quite a bit. These diseases, as I've mentioned before, with huge genetic factors, however, are actually just a couple of the examples of neurological disorders. There are many more that we will discuss in this course, but for now, it's important to put into perspective that just one of these diseases affects so many people already. And now that technologies continuously advance, we need to learn more about the root of these problems before complicated ethical dilemmas really get introduced. So now we will be diving deeper into the technologies right now, and we're essentially going to be discussing some of the basics to gene therapy. There are three major methods of gene therapies that we will discuss: gene replacement, gene editing, and utilizing antisense oligonucleotides, or ASOs for short. Let's just start off by providing some context on the gene editing and ASOs. Gene editing is the process where a DNA sequence has information altered, cut, or added, most often utilizing a Cas9 enzyme to break apart the DNA. This allows some sort of complex biomolecule to make edits on the DNA, most famously CRISPR. Now, ASOs are something a little less known because they're a more temporary procedure that can bind and modify messenger RNA in order to regulate the production of proteins. We'll go more into depth about CRISPR and ASOs in a later video. Now, let's discuss gene replacement, where essentially we are replacing a dysfunctioning gene with a healthy one, hence the name. This method most often utilizes vectors or harmless viruses to carry a healthy gene and then transport that information into another cell or organism. There are two main types of vectors we will discuss. However, there are many more with their own specialized properties. The first one, which is utilized in a ton of research, is an AAV vector, or an adeno-associated virus vector. These vectors have very low immunogenicity, meaning that they do not provoke an intense immune response once implemented. It has been used to treat cystic fibrosis, hemophilia, and is currently being researched for Alzheimer's and Parkinson's too. Aside from vectors from actual viruses like AAV, there are also things known as non-viral vectors, which are essentially complicated compounds, either natural or synthetic, that carry genetic information into the cell, for example, lipids. This is deemed to be safer, however, it has a more temporary effect as seen in research. So the last thing that we will be discussing for today is how all of this core information, particularly with the vectors, can be applied to something as selective as the brain. So for some context, we have something called the brain-blood barrier, or the BBB, which is essentially a layer of endothelial cells that behaves like a filter to only let in specific nutrients and molecules. One can assume that something as biologically complicated and traditionally dangerous as a virus for most vectors shouldn't be let in. But how is it? We can see in much research with mice specifically that it is possible. Like how we are able to modify genes with gene editing, we can also modify the vectors themselves so that they contain binding receptors that can link with the recognition receptors of the BBB and therefore pass through via something called transcytosis. Really interesting and meticulous stuff here, and I thought it was interesting how we could sort of apply this really complicated core information about gene replacement and apply this to something again as complicated as the brain. Anyways, that was it for this video. It was a bit of a long one. Thank you so much for watching, and feel free to move on to the next episode to learn more. Thank you.